I haven't had any role in the development of any chips, but I'm much from the side of the user, and I've very rapidly become a heavy user of the Affymetrix chip. So my name's Andy Breakspear, and I first of all want to thank the organisers of this session, Affymetrix, in particular Stan Trask, for giving me this opportunity to come and present our data. And the title of the talk today is The Root Hair Infectome of Medicago Truncatula, Secrets of Hair Removal. So this is the beast that we work with in the lab, Medicago truncatula. It's a legume that's native to the Mediterranean. But most interestingly to me is that it's got a couple of really cool tricks up its sleeve. Okay, so Medicago actually enjoys a great relationship with at least two microbes, okay, namely the bacterium Rhizobium meliloti and the fungus Rhizobia irregularis. And each of these microbes provides the plant with a source of nutrients that it wouldn't ordinarily be able to get on its own. So in the case of the bacterium, this supplies a usable source of fixed nitrogen to the plant. And in the case of the fungus, this supplies a source of phosphorus to the plant. In return for these goodies, Medicago feeds these microbes sugars which it derives from photosynthesis. Now, the bacterial interaction takes place in something called root nodules. These are specialized organelles of the root. And the interaction, the exchange in nutrients takes place inside these nodules. And we term this the root nodule symbiosis. And the fungal interaction takes place in specialized cells known as arbuscules. These are contained within the cortex of the root. And we know this interaction as the arbuscular mycorrhizal symbiosis. So the arbuscular mycorrhizal symbiosis is found within 80 to 90% of land plants, so a lot of plants are able to perform this trick. However, with the bacterial symbiosis, it's mainly restricted to legumes. Okay, so actually having a source of nitrogen on tap is real bragging rights for these legumes and Medicago. And the ability to participate in this symbiosis is of real interest to us. So briefly, when these two organisms meet in the soil, the root and the bacteria, they first of all want to check each other's credentials. They want to see whether they like each other. They don't want to live with each other. They might not get on. So the plant exudes flavonoids into the soil, which these are recognized by the Rhizobia. And if they like those flavonoids on offer, then they synthesize something called nod factor or nodulation factor. The plant then recognizes the nod factor, and like the bacteria, if it likes what's on offer, then it will initiate a program to accommodate the microbe. And how the bacteria gets into the plant is the most remarkable process that you'd never imagine. Okay, so as they check each other out and they like each other, the bacteria stick onto the tips of the root hair. Okay, the root hair then starts to curl around those bacteria and they grab it tightly. And this is what you can see in this image on the left. This is a microcolony, and the blue staining is the bacteria. At some point shortly after that, the plant starts to build a tunnel-like structure that we term an infection thread. And this tunnel, this infection thread, bores down the length of the root hair and into the cortex. Okay? And essentially what this infection thread does is it provides safe passage for these bacteria to get into the cortex and the nodule primordium, which is the site of nutrient exchange. And then a little later on, you can see the path of these bacteria. They've made their way into the nodule primordia, and we're starting to get deformation of the root here. And a few days after, we have the fully mature nodules here. Now, over the last 20 years or so, many of the genes which are required for this process have been determined and assembled into a pathway which we know as the symbiosis signaling pathway. So briefly, I said earlier the Rhizobia produce a nod factor which is recognized by plant receptors. This then requires the actions of DOMI1 and 2 to generate a calcium spiking signature. This is perceived by these two proteins, DOMI3 and IPD3. This then activates a set of GRAS transcription factors, and then we get activation of more transcription factors, and finally we get the infection and nodulation. So we call this set of genes here the common symbiotic components, and this is because the same machinery here is also used for the arbuscular mycorrhizal symbiosis. So in a very similar fashion, MIC factors are recognized by the plant receptor. It then goes through this, through these common symbiotic genes and back out the other side with a different combination of transcription factors, which ultimately leads to accommodation of the fungus. And many of these components have been identified using traditional forward genetics techniques, and 3 examples are NIN. This can't make any infection threads, so it can't deliver those bacteria to the growing nodule. ERN1 forms these branched infection threads, which never really make it to the base of the root hair. And likewise, the NFYA1, these form aberrant infection threads too. And here they are in the pathway. So, although we know many of the genes required in the signaling pathway, the processes that go on, the molecular processes that go on to get these infection threads initiated and extending are really poorly understood. And so it was our goal to improve our understanding of this. And what we did was to use the Affymetrix Medicago truncatula genome array. And we're particularly interested in these infection-specific transcripts in the root hair. And so we argued, if we take the whole root, then transcripts in the cortex of the root are going to dilute these transcripts, these infection-specific transcripts in the root hair. And so we decided to take pure populations of root hair and use this material to probe the array. So that obviously poses the question how to get these root hair populations. And while we briefly considered conventional methods of human hair removal, it was obvious that these weren't going to be successful. And unfortunately, the method that works very well is plunge freezing and paintbrush fracture. And so if there's anyone in the audience that's not familiar with this technique, it basically involves taking a loaf tin here, filling it with liquid nitrogen, taking your plant, plunge freezing, taking a paintbrush, scrubbing off those root hairs, and basically repeating 150 times. And so then you'll have a trough full of your root hair. You then pour this liquid nitrogen into a tube, the liquid nitrogen boils off, and you're left with a pellet of root hair at the bottom. Oops, wrong button. So this is an example of a particularly well-shaved root. You can see the stubble here. And then if you look into the pellet at the bottom of the tube, then you see this rather fragmented root hair. And if we look more carefully, you can actually see the infection inside these harvested root hairs. So we really felt confident that we were getting the material that we were looking for. And in terms of RNA, it was very straightforward. It took 150 plants to yield about a microgram of total RNA. 150 nanograms was labeled, and as is usual with these experiments, 3 biological replicates were performed for each treatment. Now, when my supervisor Jeremy and I sat down at the start of this project, and we both talked about these elongating infection threads and thought, you know, we really need to learn more about this. And I went away and started planning the experiment, and a few days later Jeremy's calling me up. Andy, actually, have you thought about looking at microcolonies? We could think about what's required for the formation of microcolonies. And Andy, have you thought about the hairs before they show any physical signs of deformation? And so we went ahead and planned some more experiments And the story continues. So we wanted to explore the effects of purified nod factor on the root hair. And also we took a mutant sickle. So this is a hyper-infected mutant, so it forms about 10 times the number of infections than a wild type. So our rationale was, well, if an infection-specific gene should be present, the transcript should be more abundant in this hyper-infected mutant, so we should be able to detect them with more sensitivity. And then finally, a set of transcription factors. These are key components of the signaling pathway that I talked about before, and we aim to find out the targets of these. So this comes to about 5,850 brushed roots, and that's using 39 of the Affy gene chip arrays. And you know, when you do these types of experiments and your heart's beating when you get the results, because you put a lot of effort into this. And I remember scrolling down first and seeing some of these known infection genes. And this first column is the degree of upregulation of these already characterized genes. So it was a relief to see these. And then this is the sickle mutant, the hyperinfected mutant. And you can see that the degree of upregulation in each case is actually higher. So we got what we were expecting, what we wanted to achieve. Now, the Affy chip had been used previously to look at infection, back by Benedito et al. In 2008. And this group used whole roots, and they found just over 1,300 upregulated transcripts. And when we compared our closest time point, the 5 dpi root hair, with the elongating threads, we saw much fewer genes upregulated. But when you actually compare these lists of genes, there's 109 core, this is all normalized together, 109 core genes, but we actually get 230 new infection-related genes from looking at the root hair. So this we wanted to narrow down. So what we did was to take the transcripts which were really specific to those infected hair. So we wanted things which were very low or basal or switched off in uninfected hair, but then are switched on on very high levels during infection. And so these are a subset of those genes. So this is a heat map, and I guess most people are probably familiar with these, but if you're not, these are probe sets down the left here. They represent genes, and I've annotated a few of them because we know the names of these genes. And if you read across, then you can look at the expression levels in the experiments that you're interested in. And so that the colors, the blue here is a very low or basal level, and the red is a very high level of expression. So to take this first BCP1 as an example, 1 dpi in an uninfected control, it's pretty much off, or it's at basal levels. It's sharply upregulated during infection, and the same is true at 3 dpi, very sharp upregulation during infection, and also at 5 dpi. And then if we add in our hyper-infected sickle mutant, again we're seeing this sharp regulation, and then finally in the nod factor experiment, we're also seeing in the nod factor control very low levels going to, in general, high levels after nod factor treatment. So we then added to the normalization of the previously published whole root data, and you can see that these guys don't even register a blip in this experiment. So actually looking and taking the hair really helped. We also brought the previously published mycorrhizal data into this. And so the first column is a control roots that don't have the fungal mycelia, and then the last column are mycorrhizal roots with the fungus. And so what we have here are two panels. The lower panel are genes which are not upregulated in the mycorrhizal symbiosis, but are upregulated in most of these experiments in the root hair. And so what we have here are new rhizobial infection candidates. And then on the top panel, we've got genes which are upregulated in mycorrhizal symbiosis. so potentially involved in the mycorrhizal symbiosis, and also upregulated in the root hair in the bacterial symbiosis. So this is really interesting, because the previous data using hair roots had suggested that these genes were mycorrhizal specific. But as it turns out, there are potentially new common symbiotic candidates. Remember, I mentioned that we have these common symbiotic components, and so what we have here are potential genes which are involved in both these symbioses. So now I'm going to slightly switch gears to more of a functional analysis of the data, and with reference here to the developmental time course, 1 DPI 3 and 5. And what we did was to take all genes which were upregulated by at least twofold. And as is usual with these types of things, we did a functional survey and many functional classes were represented. But what grabbed us was the hormone-related category. So hormones have not been associated with infection in the root hair in Medicago previously. So this was really exciting. And to expand this, you can see that the signatures for auxin, strigolactone, racinosteroid, gibberellic acid, and cytokinin are all represented. I want to talk a little bit more about auxin. So the auxin genes that are regulated are shown here: SAO1, GH3, ARF16A, and IAA9, and these are all early auxin responsive genes. And if you look in the dark blue bars, this shows the level of upregulation of these at 5 DPI, so during infection thread elongation in a wild type. If we look at the lighter blue, this is the hyperinfected sickle, and you can see that we're getting an extra degree of upregulation. And remember that this kind of fits our model for infection-related genes. They're up in the wild type, but they show a higher degree of upregulation in the mutant. And then 2 of them are also upregulated by NOD factors. So we wanted to validate our data on the array, and we checked many genes by qPCR, and that included these 4 auxin-responsive genes. And basically, the qPCR mirrored the data that we were getting from the array, so we're very pleased with that. So as I said earlier, that the goal was to find these infection-specific transcripts in the root hair. And so what we did was we checked the expression we actually used all of the auxin-responsive genes, but I'm only displaying the ARF16A here. And we did promoter Gus analysis to look for the location of the expression of this auxin-responsive gene. And indeed, we were really excited to see and a little bit relieved that yes, this particular gene is expressed in infected root hairs. And here you can see a little bit later on these patches of expression where you find the infections. And then this is ARF16 a little bit later on in the developing nodule. You can see there's a lot of expression actually within that nodule. And then this is restricted to the apex of the nodule later on. So we then obtained 3 null alleles of ARF16A, 1, 2, and 3 here. And the blue bar shows the number of infection events, and this is the wild type, and these are the mutant alleles. So you can see that in each case, there's significantly less infections present. So we're really pleased to see this. So finally, I just want to talk a little bit about these transcription factors and the profiling that we did with these. So there's a few targets known for NIN, but other than that, these are, it's really unknown what these guys are controlling. And if we want to understand infection, then we need to understand what these guys are doing. And so first of all, with NIN, this is another heat map. This first column is 5 DPI control routes. So the upper part here is mainly very low levels. And then this is during infection. These are all upregulated genes. And then if we take the mutant at the same time point, you can see that this upregulation of these genes is pretty much abolished for a large proportion. So it turns out that NIN seems to either regulate directly or indirectly a large amount of infection-related genes. So this is very exciting to us. And then similarly for ERN1, we have these genes identified here, but there's a much fewer genes here, and then for NFYA1, fewer still. We then constructed a gene regulatory network using the server at the Noble Foundation. So here we have NIN, transcription factor ERN1 and NFYA1. The nodes around here are the probe sets, which are correlated with these transcription factors. And the red color are genes or probe sets which are down-regulated significantly, at least twofold in NIN. The purple are down at least twofold in ERN, and the green one is significantly down in NFYA1. And we were pleased to see that those known targets of NIN that have been previously published were all in this analysis. And then what we've done now is to pick out a bunch of other genes which we're interested in, and we're currently trying to validate these using CHIP PCR. So that just leads me to summarize what I've said today. The transcriptome of truncatula root hair cells during infection with S. meliloti has identified new rhizobial infection and common symbiotic candidates. A role for auxin signaling during infection has been revealed, and transcriptional profiling of root hairs has revealed potential targets for key symbiotic transcription factors NIN and NFYA1. So I want to finish by asking myself a question. If I was offered the chance to start this again, firstly I'd hire an army of people to help me with the brushing, but secondly I'd pick up the new version of the Affymetrix Medicago chip, which is out this year. This has been constructed with the guidance of many people from the Medicago community. It's based on the latest annotation, so this version is much more complete than the previous version. And it also has, a real bonus for me is that it also has the ecotype M. truncatula R108. So this is becoming used a lot more in the community, and to have that on the chip is a real boon, I think. And then also Medicago sativa is present there too. So if you haven't had a chance, please go and check out this poster on the new chip later. So that just leaves me to thank, first of all, my supervisor, Jeremy Murray. He was really a great boss. He gave me a lot of freedom, as long as I did the chips he wanted to. And so he's really cool. And Chung-Woo Liu, he's done the promoter Gus and some of the functional analysis with me. And Nicholas Stacey came along at a time when I was really struggling with the root hair. We were having a lot of problems. And when you've got two people troubleshooting, it really helps. It's really good for morale. So thanks. And also thanks for you guys for listening.